So humans harbor trillions of bacteria. Most of the bacteria in the human body are found in the gut, where they're performing lots of really important functions for us. So they help process the diet, they train our immune systems, they regulate our development, and there's actually even evidence accumulating now that they influence our mood and our behavior. So these observations raise a natural question: where did all these bacteria come from? Or put a more sophisticated way, what are the evolutionary origins of the bacterial lineages that populate the microbiome? So when we were thinking about how to answer this question, we realized that our closest relatives, the great apes, may harbor some clues about the evolutionary origins of human gut bacteria. So we obtained fecal samples from African apes residing throughout equatorial Africa. So we have gorillas in Cameroon, bonobos in the DRC, and chimpanzees in Gambia, Tanzania. And we sequenced the bacterial communities present in these fecal samples. And in particular, we used a new method that profiles the strain level variation in the microbiome that's not normally seen by the standard 16S analyses. And this is called phylotagging, and it's a gyrase B amplicon sequencing approach. And there's the citation if anyone wants to read more about it. And today I'm going to focus on the results from 3 bacterial families that we've investigated so far: the Bacteroideaceae, the Bifidobacteriaceae, and the Lactospiraceae, which are 3 of the most abundant bacterial families in the human gut. So using the gyrase B data, I built phylogenies for these 3 families to try to figure out what are the origins of human gut bacteria. And what we found is that hominid speciation occurred in concert with the parallel speciation of multiple lineages of gut bacteria. So in these figures, each branch represents a bacterial lineage, and the branches are colored based on the host species from which they were recovered. And you can see that for all of the Bacteroideaceae and Bifidobacteriaceae lineages, all the relationships reflect a history of co-speciation with hominids, except for one instance where it looks like a Bifidobacteriaceae lineage has moved from bonobos into gorillas. And you can see in A and B that it looks like in 2 of the Bacteroideaceae clades, the human lineage has been lost, but the 2 clades that have human lineages those human lineages are some of the most abundant lineages in the human gut, things like Bacteroides and Bifidobacterium. So this result indicates that many of the most predominant human gut bacteria are ancient, and that they've been passed down from generation to generation for millions of years, just like our genes have been. So we can also estimate the divergence times using BEAST of the co-speciating Bifidobacteriaceae and Bacteroideaceae lineages that we recovered. And we find that they're in broad agreement with the divergence times of the hosts based on fossil and genomic data. So the human-gorilla split has been somewhat contentious historically. And so I think bacterial data can maybe provide a new line of evidence into dating host divergence times. And our data suggests a pretty early divergence time between humans and gorillas, a little over 15 million years ago, which is in agreement with some more recent studies as well. But it's important to note that not all bacterial families, I think Naomi mentioned this as well, that not all of the bacteria are co-speciating. So many of the lineages seem to move frequently across host species boundaries. So the Lactospiraceae, if you look at this tree, it's all the colors are all jumbled up. So bacteria are frequently moving across host species boundaries. So this indicates the microbiome is a composite of both co-speciating and then also independently diversifying bacterial lineages. So I think one of the interesting future directions that is suggested by this work is the question of how deep does this co-speciation go? It's possible that all vertebrates have diversified alongside some shared ancestral bacterial symbionts, which would be, I think, really interesting. So I'm currently working from the Museum of Vertebrate Zoology at UC Berkeley and collaborating with lots of good, great biologists who have really active field programs. And we're trying to do a microbial survey of the vertebrate phylogeny to hopefully better understand how we've co-evolved with our bacteria. So thanks for listening.